guys, welcome back. Today we're going to be looking at the second part of our functional genomics unit. So in the first unit, we looked at using some in silico approaches to this reverse genetics idea of going from gene to phenotype. We looked at a few other ways to kind of explore functional genomics using microarrays and other types of chip technology. So today what we're going to be looking at is another kind of reverse genetics technique called making a knockout. So a knockout effectively means that we're going to be intentionally breaking a gene of interest. So you guys know that a big theme in genetics is breaking things and seeing what happens. So we have looked at this quite a bit when we talked about the Neurospora experiment from Beadle and Tatum, thinking about mutagenizing the Neurospora. We've mutagenized some other things and talked about that. Creating this phenotype, that's what the deal is. So with a knockout, this is a reverse genetics approach because we're going to be going from the gene that we're interested in to trying to figure out more about how it's going to be causing the phenotype. So if we break it, we're able to observe that phenotype more clearly. So today we're going to be looking at type of knockout creation that's going to be achieved via homologous recombination. So that's going to be that same homologous recombination that you may remember from just looking at meiosis. Okay, meiosis 1, homologous chromosomes coming together. Okay, this is going to be a similar idea, but this time we're going to be capitalizing on the cell's willingness to do this to get it to accept some of the modifications we're going to add to it. Okay, so we're going to look at an example in yeast. So it may not seem like yeast would be the most exciting thing to work with, but in this case it's going to be pretty helpful just because yeast are going to be dealing with one cell at a time. We won't have to worry about making modifications in a whole organism at once. Okay, so let's take a look at some of our equipment. All right, so you can see in the top, we've got recombinant construct, okay? These constructs, we'll be talking about this in a lot more detail in the next video, but for now you can kind of think about it as a plasmid, okay? This is kind of the linear version of just the bottom down here, okay? I've kind of color-coded it, but really you could think about this being part of that plasmid, kind of a big circle, okay? At the bottom, we've got our chromosomal gene. So this would be in the same cell. These guys would be in the same vicinity for this to work. So we'll be looking at that a little bit later. Okay, so let's explore some of the elements that we've got going up here. Okay, so we have kind of some regulatory elements. Okay, so transcription start, regulatory elements. Okay, all of this green stuff that we see here, this is just going to be plasmid business. This sort of construct business that we don't need to worry about right now. And think about the transcriptional start site. That's going to be important because once we get it into that organism, we want there to be some expression, okay? We've also got, right in the middle here, a selectable marker, okay? So this particular one is for kanamycin resistance. This is going to be antibiotic resistance, so this will be important for keeping the cells alive if they have this selectable marker. Anything that doesn't have this, when we plate it on our special media that's going to have kanamycin, this is going to be gone. Okay, so these cells are going to be gone. So basically we need to make sure that we have this inserted or the cells will die. So we'll check on that again in a bit. Okay, and some of this sort of area just adjacent. Okay, so part of this green part that would be just adjacent to our transcriptional regulatory elements, not necessarily all this part up here, but just some of the stuff going on directly adjacent, is going to have to match what's going on with our chromosomal gene, okay? And by match, I mean it needs to have the same sequence. If you think about how homologous recombination happens in meiosis, how those chromosomes find each other, it's based off of sequence similarity. So again, homologous chromosomes, they have basically the same genes on them. Okay, they have equivalent genes with some minor differences. Overall, a lot of sequence similarity. They are not identical, but very nearly so in terms of the types of genes they have on them. we have to set up that same scenario artificially here. Okay, so we have a matching flaking sequence. All right, so let's go ahead and look at what's going to happen next. Okay, so we'll zoom in on this matching flaking sequence so we can actually see what this would look like. Okay, and you can see that if this is part of our construct, and you can see I've just kind of omitted the middle here so we can focus on the two ends. Okay, you can see we're going to have the same sequence, our ATGCC, etc. The same sequence here. Okay, it's just a double-stranded. And we have at the 3' end of this, you can see that this is going to be the same sequence. So we have to have at least a region that will be similar. Okay, so probably needs to be a little bit longer than this, but this is just kind of an example. So if we kind of zoom back out and think about what's about to happen here, hopefully you kind of remember that during homologous recombination, we're going to see kind of a mix and match. We're going to see the situation where we're kind of swapping out pieces of the chromosome. Okay, so we can kind of see what's going to happen here, right? Get those guys out of the way. And we're going to be physically switching this out. Okay, so we don't really care about the rest of this construct. We can kind of leave that alone. But what's important is that this whole bit has now integrated into the actual original gene. You can still see kind of the ends of it here. I made this kind of a slightly different color just so we can see what's happened. Okay, so two things are really important here. This is a knockout because we've broken the original gene. That original gene is no longer viable. Okay, the second important thing is that now we have that selectable marker. So again, this is going to express a protein that's involved in making these guys resistant to antibiotic. So what we're going to see is cells that have this kanamycin resistance gene. When we plate these out on media with kanamycin, they're going to live. So our transformed cells are going to live, but our cells that did not accept this or homologous recombination didn't occur, we're not going to be able to see those guys live because they don't have any kind of way to defend against our selective media. Okay, so we have to have that happen. That's just a check for us. We've broken the gene. It doesn't really matter to us how, but we've conveniently kind of embedded this expression of our kanamycin resistance here. Okay, so let's look at another way that we can confirm that we have successfully been able to get this knockout to happen. Okay, so if we have an unsuccessful deletion in our original gene, which is still going to be represented by this big long piece, okay, we can actually use a PCR technique with primers. Okay, we can put some at this end, and if we can get something amplified from this original gene, we can do it on the other end too. If we get this product, okay, notice that we are going to have these sort of internal primers. Okay, if we are able to amplify that, that's bad biscuits. Okay, by contrast, we can use another PCR technique using the same kind of outer primer. That can be the same, but now we've got one that matches the transcriptional regulatory business that was going on with our construct. Okay, we do the same on the other end, part of that kanamycin resistance gene. Remember, these primers have to match what's going on here. Okay, so if we can amplify this, we've probably done a pretty good job of getting that construct to drop down into the chromosomal gene. Okay, honestly, in the real world, we probably also sequence this first just to make sure that it looks correct, but this would be a pretty quick check to do before you started all the work that was involved with sequencing. So that's one of the things that we can do. Okay, so we've looked at this example in yeast. Again, that's kind of a simple model for us to think about because we're not trying to, again, put this into an entire organism. Okay, so when we do try to put this into an entire organism, we have a couple more things to consider. We have to get this knockout in all of the cells of the organism. Okay, so we'll be looking at how to do that. in mice. Okay, in mice, obviously, this is a really important animal model system that we use a lot of times for modeling what's going to happen in disease states, for medicine, that type of research. So it's important for us to understand what's going to happen. Okay, so let's look at our construct that we would use for these type of cells. Okay, so there's a lot of stuff that's similar going on here, a lot of similar things going on here. Okay, so this is going to be, again, our recombinant construct that we make in the lab. All right, our chromosomal gene that's present in mice. Okay, so you can see we've got, again, similar things. We've got the ends, the flanking sequences that will match that target gene. Okay, we have another selectable marker. This time it's neomycin, but it's going to perform the same function as our kenmycin resistance. Okay, you may notice we have an extra little player over here, another character. This is a little coding unit for an enzyme called thymidine kinase. So that's going to help us make sure that we've done everything correctly. Okay, so what we're going to do is take this construct that we've hopefully made, okay, and we can mix it with an agouti mouse stem cell. So this is an embryonic stem cell from an agouti mouse, okay? So agouti is just a fur color. This is what these guys look like. So they're a nice golden color. This guy is a little bit thick, but that doesn't have anything to do with what we're going to do next, okay? And it is fine to be a little bit thick. It's okay to be that way, okay? So this agouti mouse, the color of this guy, is going to help us later on. So I'll be reminding you about this agouti mouse a little bit later. We're also going to be kind of adding in some black mice as well. Okay, so what we're going to do is take embryonic stem cells from a mouse that was going to be agouti. Okay, so these again are going to be just one cell embryonic stem cells. And once we've done that, either we will have correct homologous recombination, which will disrupt the target gene, just as we saw in our yeast example, or we can see some off-site integration, leaving the gene intact, okay? It's true that that could happen in yeast as well, but again, yeast are single cell, so we can, you know, get a huge population of those guys, and using our PCR validation, get a pretty good idea of what we're dealing with here. However, one of the problems with mice is that they take a really long time to get to the point where we can actually study this. Okay, so we're going to see that we're going to have some breeding involved. We're going to have to grow this up into a mouse. There's just a lot of time involved with this. So you really don't want to mess this up at the beginning. So we're going to have a little bit of extra insurance for this. And that's going to be in the form of our thymidine kinase. So we will see how that works. So let's take a look at what happens when we have correct targeted insertion. Okay, so here is going to be our construct. Okay, our correct targeted instruction, homologous recombination occurs. And notice in the chromosomal gene, we do sort of now we have collected our neomycin resistance, so that's good. We're going to be able to survive that selective media. But notice we've still left this thymidine kinase behind. That's good news right now, okay? So let's see why that is by looking at the situation where we have random, not targeted insertion. Okay, so in this case, what we've seen is that the chromosomal gene, this could be like an off-target place, okay? Because we are off-target, we are actually nowhere near the original gene in the chromosome, okay? You can see that again. We've actually done an off-target sort of switch, okay? So the original yellow gene in the chromosome was not in this picture. There is no incentive to recombine using these flanking sequences, okay? Because this light blue portion didn't really match the original chromosome area that we're working in. But this yellow part that should be flanking our targeting sequence, that didn't match just as much as any of the rest of this. So there's no real incentive to recombine specifically here and here. So what that means is that generally we will get this sort of incorporation of thymidine kinase. Okay, so in a random insertion, we're going to see that thymidine kinase can be expressed. So let's compare these. We've got our correct insertion. Here's the original chromosomal gene. We've disrupted it, but we do have our neomycin resistance. In the non-targeted example, our original gene location is somewhere else. Okay, I've kind of noted that by kind of breaking this. This could be on a completely different chromosome. It can be somewhere else, but we have not targeted that gene. Instead, we've just dropped that chunk from our construct somewhere else. I don't know where. But again, there's no incentive to recombine in these flanking sequences, so we've dropped this thymidine kinase in as well. Okay, so to see what happens with this thymidine kinase, let's put these into mouse cells. Okay, so we're going to go ahead, put them into our cells and see what happens. Okay, so here's our two cells, one with the correct scenario, one with the not correct scenario, remembering that the original gene is still intact somewhere else that it's not pictured. We're going to plate them on media that contains neomycin, which is going to be our actual selection, and ganciclovir. Okay, so ganciclovir is going to help us out with these incorrect cells. Okay, so let's deal with the first correct situation first. These cells are going to be resistant to neomycin, and they don't have any thymidine kinase to express. Okay, the relationship between thymidine kinase and ganciclovir is such that if you create this enzyme, this thymidine kinase enzyme, in the presence of ganciclovir, that will kill you. Okay, so in the incorrect situation, technically they would be probably resistant to neomycin, depending on the sort of transcriptional machinery around wherever this has been dropped. But because they are going to be expressing this thymidine kinase on plates that contain ganciclovir, they will end up dead. So what we're left with are only cells that are likely to have the correct placement of this cassette. Okay, so of this piece of this construct, this is going to be what we're looking for. Okay, so let's actually see the next steps. Again, moving forward with only the correct cells. The thymidine kinase has saved us a lot of time. Okay, so let's see what happens. We're going to take these cells that now contain the knockout, remembering that they were from an agouti mouse. So I've kind of color-coded it so it's a little bit more yellow. This is from an agouti mouse, okay? We're going to put it into a blastocyst from a black mouse, okay? So a blastocyst, this is going to be one of the very early stages of development, okay, when we're developing from a single cell zygote into an actual organism. One of the early stages is a blastocyst, okay? This is going to be our blastocyst. It's going to have, again, it's going to be made of cells, sort of outer layer. But the inner cell mass, that's going to be kind of what we're targeting. This will be the part that's actually one day going to become a mouse, okay? And so we've got now our black mouse. We take a blastocyst from it. It's started to develop. And we just drop this into our blastocyst, okay? So there they go. And we let it kind of keep dividing, okay? So this is just like kind of like random, sort of keep dividing, okay? And you can see that some of the cells that came from our agouti mouse that have the knockout, they're going to divide, and then we're going to have cells from this black mouse as well, because this already contained a developing sort of mouse cell situation, okay? So the next part of this is going to be really important. Again, it was important to use these contrasting colored mice okay, because they're going to help us out to understand what's happening. So let's go ahead and pause that video. And what do you think the color will be of these types of mice that come out of this situation? Okay, hopefully you came up with at least some type of mix of agouti and black. Okay, so this mouse is what's called a chimera. So you can see that it's got kind of black patches, kind of like some agouti patches in here, some light and dark colors, okay? This happens because part of its cells are from that knockout that we created, that agouti cell line, and some of them are from the black mouse from which we got the blastocysts, okay? So this is okay, but remember that our goal is that we need to have this knockout in all the cells of the mouse before we get ready to analyze what this gene is doing, okay? So what we're going to do is cross our fingers that some of the germline cells in this mouse, some of the cells that are going to produce gametes are going to be from that agouti knockout cell line, okay? Then what we're going to do after crossing our fingers, if that's the case, is we're going to start mating this guy so we can get a mouse that has all knockout cells, okay? So we make this with a black mouse, and what we should get, again, if we do have kind of some of the gametes will arise from the agouti mouse, the container knockout. Some of them will arise maybe from the black mouse, okay, that will not contain any knockout. So they've got the wild type gene, okay? The agouti mice that come out of this, they're going to be heterozygous, all right? And that's not good enough because if we really want to know what that phenotype is, we have to get this to be homozygous, okay? So we keep reading this. All right, we mate siblings until we get an agouti mouse that is going to contain knocked out copies of this gene for both chromosomes, okay? So we have now homozygous knockout. Once we get to that point, now we can study this guy. So you can see why, after all of this time, we would want to be careful that we have done this correctly in the very beginning steps using those selectable markers and thymidine kinase. Okay, so making knockouts, this is, again, kind of the gold standard for illustrating what phenotype a gene is going to cause. Whatever we've knocked out in this mouse, we will hopefully see some type of phenotype associated with it so that we know this is what this gene is doing, because when we remove it, that job isn't done anymore. Okay, so we can think about some conclusions to take from this. Okay, we can create these knockouts. This is going to be a reverse genetics approach to explore gene function. This would be used when we determine that a gene is going to be something we want to study. We want to make sure to validate what phenotype that gene is responsible for, okay? And we do this using very careful planned construct design, making sure that we're going to be able to target what we want to target, along with homologous recombination, which the cells will naturally want to do if we get this construct created correctly, okay? And so next time, we'll be learning more about how these constructs work. We're going to be learning more about how to make them, how to check if they're correct, connecting this up with some of the stuff that we did with mice today, and maybe even some of the stuff that you guys saw in the video about CRISPR. So go ahead and end here with our biology joke of the day.